Welcome back to Biointron's Antibody ABCs. What is next-generation sequencing? Next-generation sequencing, or second-generation sequencing, is a high-throughput technology used in molecular biology to rapidly sequence DNA or RNA. NGS techniques have revolutionized genomics research, enabling scientists to sequence multiple genes quickly and at a relatively low cost compared to traditional mutation detection methods. The key idea behind NGS is its massive throughput by generating millions of short DNA fragments in parallel. The most popular technology is sequencing by synthesis, which was leveraged by Illumina to produce approximately 90% of the current world's sequence data. This approach relies on 4 fluorescently labeled nucleotides to sequence millions of clusters on the flow cell surface in a parallel fashion. First-generation sequencing, or Frederick Sanger's chain termination sequencing, was developed in the 1970s. It enabled the sequencing of short DNA fragments and paved the way for automated sequencing methods, like the ABI 370, which significantly improved throughput. However, first-generation methods were time-consuming and expensive for large-scale projects, limiting their use in genome-wide studies. The leap to second-generation sequencing, also known as NGS, introduced parallel sequencing, where millions of DNA fragments could be sequenced at the same time. Platforms such as Illumina, Roche's 454, and Ion Torrent made it possible to perform whole genome sequencing, whole exome sequencing, and targeted sequencing. These methods not only improved speed, but also lowered costs, making high-throughput sequencing more accessible to researchers. Third-generation sequencing technologies, such as Pacific Biosciences and Oxford Nanopore, further improved NGS by enabling long-read sequencing. This allows researchers to sequence larger DNA fragments, up to tens of kilobases, overcoming the limitations of earlier short-read technologies. Long-read sequencing is particularly useful for genome assembly and identifying complex structural variants, such as large insertions, deletions, and duplications. Biointron focuses on antibody discovery, expression, and optimization. From gene sequence to purified antibodies, it takes just 2 weeks. Our high-throughput platform enables the rapid expression of recombinant antibodies in HEC293 or CHO cells. With our self-owned alpaca farm, we also offer a 1 alpaca for 1 project commitment, with unique binders, high diversity, and large capacity. We will meet your small-scale needs for antibodies, supernatants, and expression plasmids within 4 to 10 days. We also have a high-throughput, fully human antibody discovery platform, which will give you fully human antibodies in 3 months. Thank you for watching, and please comment with any questions or topics you'd like us to cover next.